So, for the background for Alzheimer's disease, it is thought to be caused by the abnormal buildup of proteins in and around brain cells. Alzheimer's symptoms typically begin with mild memory loss and confusion, progressing to difficulties with language, problem solving, and mood changes as cognitive functions decline. Early onset Alzheimer's is rare and can be found in 1 to 5% of cases. It's strongly genetic and often linked to mutations in APP, PSEN1, and PSEN2 genes. Late onset Alzheimer's is more common and is associated with the E4 allele of the APOE gene, which increases risk but does not guarantee development of the disease. It is estimated that the risk attributed to genetic factors is 56 to 70%, and the rest is environmental. In addition, high blood pressure, age, and long-term exposure to environmental degradation, such as air pollution, raises the risk of Alzheimer's. The proteins involved are amyloid, which are deposits of plaque around brain cells, and tau, which are deposits of form tangles within brain cells. As brain cells become affected, there's also a decrease in neurotransmitters involved in sending messages or signals between brain cells.
The 23andMe genetic test only analyzes one gene relating to Alzheimer's disease, ApoE4 or RS429358. The typical function of the APO gene is to create the protein apolyprotein E, which is vital in the creation of lipoproteins that carry lipids around the body. Each APO allele, E2, E3, and E4, synthesizes apolyprotein E, but those created by the E4 variant tend to prefer larger lipid particles. Apolyprotein E is expressed all throughout the body, mainly in the liver and the brain. Compared to those without a homozygous E4 variant, the likelihood of developing late-onset Alzheimer's disease is 12 times, and of early-onset Alzheimer's is 61 times more likely. While not entirely proven, the reason that the E4 variant is thought to cause Alzheimer's disease is thought to be an overproduction of apolyprotein E, causing protein clumps called amyloid plaques in the brain. As previously mentioned, 23andMe only tests for the ApoE4 allele. However, studies show that there could be other genes correlated to the development of Alzheimer's disease. Early onset Alzheimer's disease has also been found to be related to variants of the APP, PSEN1, or PSEN2 genes. Variants of the PSEN1 gene alone appear in 70% of cases. Similar to the hypothesized causation of ApoE4, mutation in the three aforementioned genes caused an increased buildup that forms amyloid plaques. A genetic test that tests for these three other gene variations would be able to detect the likelihood of developing Alzheimer's disease much more effectively than those provided by 23andMe.
The number of additional SNPs that impact Alzheimer's disease but aren't tested for by 23andMe is relatively large. Specifically, there's 5 SNPs that aren't tested for by 23andMe. One major variation that isn't accounted for by the 23andMe test for Alzheimer's disease is the RS75932628 SNP, which has an undeniable impact on the risk of developing Alzheimer's disease. It has a great deal of effect on brain cells, and this variant has been proven to have a large correlation with the pathogenesis of Alzheimer's disease. The gene that this SNP impacts is the TREM2 gene, which regulates the immune system and brain homeostasis. The impact of this SNP on the brain homeostasis function is what is correlated with late-onset Alzheimer's disease. A study from Iceland showed that the effect of this SNP was that anyone who had it had a 3-fold chance of developing Alzheimer's. The gene TREM2 is generally found in the microglia, which resides in the macrophages. Specifically, TREM2 to be present in the microglia that are present in the brain. Microglia serve as the immune cells of the brain, looking for any abnormal pathogens affecting the central nervous system. This is how the RS75932628 SNP has an effect on Alzheimer's disease by affecting the brain's immune capabilities. This variation was very predictive in Icelandic populations and meta-analysis, but showed no association when tested on the Chinese population.
In a study performed by S. Michi et al., getting positive genetic test results for both children and adults was shown to increase anxiety and depression in the subjects, with the range for adults increasing from a non-clinically diagnosable level to a clinically diagnosable level. Children's levels remained in the average range for a human. In another study performed by R. Sandbrick et al., it was found that mutations in the APP gene only account for a 0.3 to 5% increased risk in Alzheimer's. This differs based on what variation is in the gene. The article also mentioned that other genetic factors, not the APP or APOE genes, have yet to be confirmed and their pathophysiological mechanisms remain to be analyzed. It is worth noting that this study was performed in 1996, so other evidence might already disprove this.
Lastly, there are a few disclaimers one should know about our test. Just because certain variations that are linked with Alzheimer's, as mentioned before, appear in your gene sequence, it does not mean you will develop the disease definitively. This is especially due to certain genes like PSEN2 that have only been found to increase the risk slightly. Also, there is the limitation of the amount of genetic links to Alzheimer's disease that have not been found. As these links have not been discovered, we cannot possibly test for them. As a disclaimer, if one doesn't test positive for any of the genetic variations previously mentioned, there is still a possible risk one will develop Alzheimer's disease.